Welcome to the deep dive. Today, we are opening up the engine room of modern healthcare, the clinical laboratory.
Right.
This system is really the backbone of diagnosis. It's the place where we translate a patient's symptoms into, hard measurable data.
But even the strongest backbone needs rigorous checking.
Exactly. You need that quality control, especially when you're swimming in a sea of these highly sensitive assays and really complex instruments.
And that's our mission today. It's a deep dive, pulling that high-yield knowledge straight from a foundational text on clinical chemistry, immunology, and lab quality.
So we're distilling this massive resource down.
We are, into clear, actionable knowledge for you. We'll be focusing on the methodologies, the specific scientific how behind the tests, and maybe more importantly, the critical errors that can happen.
The pitfalls.
The true clinical pitfalls, yeah, and the real-world context of major body systems.
We really want you to walk away understanding not just what a result means, but how reliable that number is and what might, compromise it.
A thorough and efficient learning experience. That's the goal.
So let's unpack this. Where do we start?
Let's start at the very foundations. The analytical instruments themselves. The majority of routine measurements you'll encounter in the lab rely on, basic physics and light.
Spectrophotometry.
Spectrophotometry. It's the most common method of analysis. You're essentially just quantifying something by measuring how much light it absorbs.
Or sometimes how much it transmits, right?
Exactly. And that all relies directly on Beer's law.
Ah, Beer's law, that beautiful linear relationship.
It is the concentration of the substance and the light it absorbs. If the concentration doubles, the absorbance doubles. That's simple as. But of course, the clinical lab needs a lot more than just colorimetry.
Right. We have urgent measurements, things like electrolytes and blood gases.
For those, we rely heavily on highly specific chemical sensors. So think of the ion-selective electrodes that are essential for measuring sodium and potassium.
The quick, precise measurements for a basic metabolic panel.
Exactly. Or the electrodes used in blood gas analyzers, which function as, micro pH meters and oxygen sensors.
But for the most sensitive and definitive results, we have to move into the heavy artillery.
Chromatography and mass spectrometry.
Right. Why do we consider methods like GCMS and LC-MS the gold standard?
Well, because they offer superior specificity.
Right.
And they're robust. They physically separate compounds before identifying them chemically.
Okay.
Liquid chromatography tandem mass spectrometry, LC-MS, is often the single most sensitive method we have in a clinical lab.
And that's essential for things like, what? Therapeutic drug monitoring.
Therapeutic drug monitoring and definitively confirming drugs of abuse. Places where you need to separate and identify a very specific molecular fingerprint.
Speaking of complex molecules, the immunoassays are they're critical to the modern lab.
Absolutely. They let us measure things in the pico and nanomolar range.
But their reliance on antibody binding is where you get these unique risks for interference. So let's quickly define the two main design types.
So immunoassays generally use one of two broad strategies. First, you have the competition immunoassay.
Okay.
This uses one antibody, and it's typically designed for small molecules, often drugs, where the patient's analyte competes with a labeled reagent for a limited number of binding sites.
And the other, more complex system, the one for larger molecules.
That's the immunometric, or, as it's more commonly known, the sandwich immunoassay.
The sandwich.
It requires two separate antibodies that recognize different parts of a large analyte molecule. It's really ideal for measuring larger proteins and polypeptides, things like TSH, PSA, or certain hormones.
And here's where it gets really, really clinically critical. Because these sandwich assays are susceptible to some specific interferences that can put a clinician on the wrong path entirely. Let's start with the most dangerous one, the prozone effect.
Absolutely. And this isn't just a lab error. This is misdiagnosing a potentially life-threatening condition. The prozone, or hook effect, is a pitfall specific to sandwich assays.
So what happens?
If a patient sample has an extremely high concentration of the analyte, a classic example is a molar pregnancy with just sky-high HCG levels, that excessive analyte saturates both the capture and the detector antibodies at the same time.
And that saturation prevents the sandwich from actually forming.
Exactly. No sandwich, no signal. The assay reports a signal that corresponds to a sample with a low concentration.
So you get a falsely low or even a falsely negative result for something that's actually dangerously high.
That's the danger. It's why if a result is clinically questionable, Say, a clinician suspects very high HCG, but the result is normal. The lab has to automatically dilute the specimen and rerun it.
And dilution fixes it.
Dilution almost always resolves the issue by dropping the concentration back into the measurable range.
Okay, what about those endogenous interferences, the things that are just physically blocking the view?
Right, so we always have to watch for light-absorbing substances. The main culprits are bilirubin, which gives samples that strong yellow color.
Enteric samples.
Ecteric, yes. And hemoglobin released by hemolysis, which makes samples red. These substances absorb light right where many common assays are run,
Causing interference.
causing interference, although typically they only cause major problems when the concentrations are really high for bilirubin, usually above 20 milligdl.
And then there are the pesky heterophilic antibodies.
Ugh, the immunological nightmare.
They are.
They are. Heterophilic antibodies, which include things like human anti-mouse antibody or HAMA, are endogenous human antibodies. Their danger lies in the fact that they're just sticky and nonspecific.
So they sort of bridge the two different animal antibodies used in the kit.
That's a perfect way to put it. They bridge the gap, say, between a mouse and a goat antibody, and they form a pseudo-sandwich even if the patient's target molecule isn't there.
Which gives you a false positive.
A big false positive. Clinically, this is especially a problem in assays for large molecules like TSH or PSA. A clinician might see an extremely high TSH, diagnose hyperthyroidism, and start inappropriate therapy.
All because of interference from the patient's own immune system.
Right, not an instrument error.
Now, if the analytical phase has its pitfalls, the pre-analytical phase is a minefield.
It is. The vast majority of laboratory errors, the non-cognitive slips and lapses, happen before the specimen ever hits an analyzer.
And the single scariest pre-analytical error.
Incorrect patient identification, without a doubt. The one that keeps lab directors up at night. That's why we have to match at least two criteria like name and date of birth every single time.
But even if the ID is perfect, the collection technique is crucial. The correct order of draw is a prime example of preventing contamination. Why do we follow that specific sequence?
Because the additives in one tube can contaminate the blood collected later. So we start with microbiological cultures, which are sterile, then the royal blue tube with no additive.
Okay.
Then we proceed to the citrate tube, the light blue top, and only then do we go to the serum tubes or the gold top with the gel separator.
And that order prevents contamination from, say, anticoagulants.
Exactly. You don't want anticoagulants messing with your trace metal testing later on.
And using the wrong tube itself can just totally skew results. You mentioned lithium.
Yes. If you collect a specimen for lithium level in a tube that contains lithium heparin as the anticoagulant, which is a common green top tube, you are literally adding lithium to the sample.
Leading to a falsely elevated, dangerous result.
Directly. Another variable is patient prep. Glucose testing and lipid panels, they absolutely require the patient to fast overnight.
We also have to consider posture, which I find fascinating as a physiological variable.
It is. If a patient shifts from an upright to a supine, a lying down position, their blood volume actually shifts.
So analytes that are bound to large proteins.
Analytes bound to large proteins like renin, TSH, T4, warfarin. They can all be affected by this shift because their concentration changes relative to the plasma volume.
Okay, let's pivot from the practical errors to the stats. The quality control that ensures the lab is reliable. We have to define two core concepts, precision and accuracy.
Right. Precision is just reproducibility, how close repeat measurements are to each other. Accuracy is how close that measured value is to the true target value.
And that distinction is vital.
It is, because you can easily have an assay that is highly precise. All the results cluster tightly together, but it's consistently inaccurate because of a systematic error or a calibration drift.
We also talk about test performance in terms of sensitivity and specificity.
Absolutely essential. Sensitivity is the ability of a test to correctly identify the true positives. Specificity is the ability to correctly identify the true negatives.
So if a test correctly IDs 95 sick people out of 100 and 95 healthy people,
then that test has 95% sensitivity and 95% specificity. You need both to judge its clinical utility.
And when we talk about institutional quality goals, Six Sigma is the gold standard, isn't it?
It is. Six Sigma came from manufacturing. Motorola developed it. When you apply it to a clinical lab, the goal is extremely ambitious. An error rate of just 3.4 defects per million operations.
Wow, that's a 0.000034% error rate.
It sets the bar for near perfection.
And it's so important for our audience to understand that even normal results are statistical. They're not absolute truths.
This is a vital point that's often missed. The standard reference range covers values within the mean plus or minus 2 standard deviations. But that only encompasses 95% of a healthy population.
Which means, by definition, 5% of perfectly healthy people will fall outside that range.
Correct. And that risk is cumulative. If you run 5 independent tests on a perfectly healthy person, say, a basic metabolic panel, there's nearly a 23% chance,
23%.
22.7% to be precise, that at least one result will fall outside the established reference range.
That's why you can never treat an isolated result as definitive without the clinical context.
Exactly. The patient isn't abnormal. The statistic is just catch them in that 5% tail.
That statistical skepticism is a perfect bridge into the clinical system, starting with endocrinology. It's all about those intricate negative feedback loops.
The hypothalamic pituitary endocrine gland axes are foundational. Let's look at the thyroid. TRH stimulates TSH, TSH stimulates T4, T3 production, and then those final hormones apply negative feedback, slowing down TRH and TSH release. The system self-regulates.
And we know TSH assays can be tricky, but how does the thyroid axis impact other body systems?
Well, fundamentally, the thyroid hormone, T4, regulates overall basal metabolism.
Which includes drug metabolism in the liver?
It includes the function of the body's detoxification system, specifically the cytochrome P450 enzymes. So if a patient develops severe hypothyroidism, drug metabolism slows down drastically.
And that can lead to drug toxicity.
We've seen cases where it's exacerbated phenytoin toxicity, for example, because the drug is cleared much more slowly, leading to dangerous accumulation.
Moving down the axis to the adrenal cortex. 3 zones, 3 different classes of hormones.
Yep. Outermost layer, the zona glomerulosa, secretes mineralocorticoids like aldosterone for salt balance. The middle, the zona fasciculata, produces glucocorticoids like cortisol for stress and glucose.
And the innermost.
The zona reticularis, which produces sex steroids.
For Cushing's syndrome cortisol excess, how do we tell a pituitary problem from an adrenal tumor?
That's where the high-dose dexamethasone suppression test is key. Dexamethasone is a powerful synthetic glucocorticoid. If we give a high dose and cortisol production is suppressed, it usually points to Cushing's disease.
Meaning a pituitary tumor that's still partially responsive to feedback.
Right. If suppression doesn't occur, the cause is generally outside the pituitary, like an autonomous adrenal tumor or an ectopic ACTH-producing tumor.
And conversely, for adrenal insufficiency, we use the ACTH stimulation test.
Correct. We give ACTH, and if there's no corresponding rise in cortisol, that tells you the adrenal gland itself is failing primary hypoadrenalism.
Okay, next up, the powerhouse organ, the liver, and the critical issue of jaundice.
The liver is central to, well, countless processes: protein synthesis, metabolism, and detoxification, especially bilirubin excretion.
And when the liver's failing, we have to distinguish between unconjugated and conjugated bilirubin.
The indirect versus the direct. And this difference in solubility is the absolute key to the clinical presentation.
Especially in the urine.
Exactly. Unconjugated bilirubin is water insoluble. It's bound to albumin. It can't be filtered by the kidney, so you never see it in the urine.
But conjugated bilirubin is water-soluble.
It's water-soluble. So if it regurgitates back into the blood because of a biliary obstruction, it will be excreted in the urine, causing chloria bile in the urine. That simple fact helps localize the jaundice.
We also use that bilirubin profile to differentiate between common congenital hyperbilirubinemias.
The most common and most benign is Gilbert's syndrome. It's caused by reduced activity of the UGT enzyme that conjugates bilirubin.
So you get an isolated, elevated, unconjugated bilirubin.
Correct. In contrast, conditions like Dubin-Johnson or Rotor syndrome are from impaired excretion of conjugated bilirubin. So you see an increased conjugated bilirubin in the blood.
And as a quick toxicology example, is there one liver enzyme that acts as a strong red flag?
Yes, an isolated elevated GGT gamma-glutamyl-transferase is a classic characteristic often associated with chronic alcohol abuse, even if other LFTs are normal.
Let's shift gears for a quick review of water, electrolytes, and acid-base balance. The fundamentals, focusing on sodium, hyponatremia.
Hyponatremia, low sodium, always has to be categorized by body volume. You have absolute hyponatremia, where total body sodium is low. The patient is hypovolemic, and the body activates RAAS and ADH to conserve everything.
And then the opposite situation.
Then you have dilutional hyponatremia, where the patient is volume overloaded in conditions like CHF, renal failure, or cirrhosis, and the excess water just dilutes the sodium.
Even if total body sodium is actually normal or even high.
Precisely.
And for managing metabolic acidosis, the ending gap is paramount.
The formula is simple. Anion gap equals sodium minus the sum of chloride and bicarbonate. A high anion gap happens when bicarbonate is consumed by an acid, and the cause is memorably covered by the mnemonic muddy piles.
Mud piles. Methanol, uremia, DKA, paraldehyde, isopropanol, lactic acidosis, ethylene glycol, and salicylate. That single mnemonic is so critical in an emergency.
It really is. Now, moving to therapeutics, therapeutic drug monitoring,
Mhm.
or TDM, it's not necessary for most drugs,
Right. It's strictly for those with a narrow therapeutic index.
meaning the margin of error between a helpful dose and a harmful dose is exceptionally small.
Digoxin.
Digoxin is the textbook example. The therapeutic range is tight, 0.8 to 1.8 ng/ml, and toxicity can emerge immediately above 2.0. That narrow gap means you have to monitor it.
But when we monitor, we have to distinguish between total drug and the free fraction. Why is that so important?
Because only the unbound or free drug is pharmacologically active. It's the only part that can cross cell membranes and do its job.
So for strongly protein-bound drugs,
For strongly protein-bound drugs, phenytoin, carbamazepine, valproic acid monitoring, that free fraction becomes clinically essential in certain populations.
like patients with liver disease or low albumin.
Exactly. If a patient has hypoalbuminemia, the body can't bind the drug effectively, leading to potentially dangerous increases in the active free fraction, even if the total level looks normal.
Turning to toxicology, drugs of abuse testing relies heavily on immunoassays for screening. And as we've established, that means a major risk of false positives.
Amphetamine assays, in particular, are highly susceptible. Many over-the-counter cold medicines with ephedrine or pseudoephedrine can trigger a positive screen.
Even a Vicks inhaler.
Even the L-methamphetamine in a Vicks inhaler, yeah. Likewise, high doses of dextromethorphan from cough mixtures can cause a false positive for PCP.
Absolutely. For any legal or major clinical decision,
And people go to extraordinary lengths to cheat these tests.
they do. While routine integrity tests check for pH, creatinine, and specific gravity, sophisticated internet adulterants, potassium nitrite, glutaraldehyde, stealth, are designed to chemically interfere with the metabolites.
And routine tests can't detect those.
They often can't. You need specialized dipsticks like Adultacheck for detection.
And we also have to remember that a negative toxicology report doesn't always mean the patient is drug-free.
Correct. Exotic substances like magic mushrooms, peyote, and synthetic compounds like bath salts or spice are generally not detected by routine immunoassay screens.
The assays just aren't designed to see them.
Exactly. You need specialized chromatographic methods.
Let's look now at advanced immunodiagnostics, starting with chronic infections like HIV.
For infections like HIV and hepatitis C, the emergence of nucleic acid tests, or NATs, is crucial. NAT detects HIV RNA or DNA during that window period of infection.
Before the body has mounted an antibody response.
Right. And it's also essential for diagnosing infection in infants born to infected mothers, because those maternal antibodies will be present and confuse any standard antibody test.
What about screening for monoclonal gammopathies? We use serum protein electrophoresis, SPEP, but what's the definitive test?
SPEP is a great screening tool. It separates proteins into 5 fractions. If a localized spike is detected in the gamma region, a monoclonal protein, then immunofixation is the required definitive confirmatory test.
And that tells you the specific type.
It identifies the specific isotype, like IgG, IgA, or IgM, and the specific light chain, kappa or lambda.
And there's an important caveat about that initial screen, right?
Yes, it's crucial to remember that a normal SPEP does not rule out myeloma. Up to 11% of patients with multiple myeloma can have a normal SPEP result,
Wow.
which underscores the need for clinical suspicion, even with a normal screen.
To wrap up, we have to address an area of increasingly high clinical risk, herbal supplements.
Because they're often perceived as harmless and regulated differently, the interference risk is frequently missed.
So what are the mechanisms?
They fall into a few categories. First, direct physiological effect. Kava, for instance, is known to cause elevated liver enzymes from hepatotoxicity. Second, herb-drug interactions. St. John's wort is the classic example. It induces liver enzymes, which accelerates the metabolism of critical immunosuppressants like cyclosporine or tacrolimus.
Which, if undetected, could risk transplant rejection, a catastrophic outcome from an over-the-counter supplement.
Exactly. Third is cross-reactivity. Certain traditional Chinese herbs, like chansu, contain compounds that structurally mimic the drug we're trying to measure.
So it causes a false positive on an immunoassay.
A classic example is causing falsely elevated digoxin results. And finally, there's just straight-up contamination and adulteration.
You mean other drugs are mixed in?
We see reports of some Chinese herbs being adulterated with high-potency Western drugs like phenytoin or contaminated with heavy metals like lead, which is sometimes seen in certain Ayurvedic medicines.
That deep dive into the foundational concepts, the challenges, the hidden risks, it really gives us a comprehensive overview of a vast and detailed field.
Indeed. The final takeaway should be that the clinical lab is a precise, indispensable partner in diagnosis. But understanding its limitations, the interferences, the QC rules, the pre-analytical variables, that's just as vital as knowing the reference ranges.
You have to think beyond the number on the page.
You have to consider the entire pathway.
So let's leave you with a provocative thought that connects all these dots. Protein binding, drug monitoring, and liver function.
Yeah.
When you're monitoring a strongly protein-bound drug like phentoin, why does severe liver failure create a far greater toxic risk than it would for a weakly bound drug? And how does this dramatically change what the lab must measure?
A great question. Think about what liver disease does to protein synthesis, which affects binding, and then think about what happens to that active free drug fraction when its binding capacity is compromised.
That level of complex systems thinking is what moves you from just reading results to actually participating in the diagnosis.
Precisely.
We'll leave you to mull over that until the next deep dive.